My name is Dr. Susan Ingram from the Department of Neurological Surgery at Oregon Health and Science University, and I will be discussing my chapter on the molecular basis of nociception in this lecture. There are three main objectives of this lecture. The first is to understand the classification of primary afferents that sense nociceptive stimuli and contribute to our perception of pain. Then we will go into key molecules that are involved in sensing, transducing, and transmitting nociceptive signals. And finally, into mechanisms of sensitization that modulate proteins at all steps in the pathway from the periphery to the central nervous system. First, we're going to look at the definition of pain as defined by the International Association for the Study of Pain, IASP. The IASP has recently updated their definition of pain that was last published in 1979. Pain is defined as an unpleasant sensory and emotional experience associated with or resembling that associated with actual or potential tissue damage. The new addition to the previous definition are these 6 factors listed here that basically redefine pain as an individual experience. First, pain is always a personal experience that is influenced to varying degrees by biological, psychological, and social factors. Second, it is important to note that pain and nociception are different phenomena. Pain is a perception and cannot be inferred solely from activity in sensory neurons. Third, through their life experiences, individuals learn the concept of pain. Fourth, a person's report of an experience of pain should be respected, as each individual will process pain differently. 5, although pain usually serves an adaptive role, it may have adverse effects on function and social and psychological well-being. And finally, verbal description is only one of the several behaviors to express pain. Inability to communicate does not negate the possibility that a human or a non-human animal experiences pain. These distinctions are extremely important to understand as physicians and scientists, as they highlight the fact that pain may be caused by many factors that may not be easy to diagnose or understand. They also highlight the importance of brain processing on an individual's experience of pain. The somatosensory system encodes our sense of touch and pain. Primary afferents are neurons with cell bodies in the dorsal root ganglia that lie along the spinal cord or in the trigeminal ganglia that lies at the base of the skull. These neurons send axons that innervate the skin, viscera, deep muscle, and joints, as well as bidirectionally into the central nervous system. A beta fibers have the largest cell bodies, or soma. They are highly myelinated and are thus fast conducting. They transmit touch and vibration information. A delta fibers have medium cell bodies, are lightly myelinated, and carry pain and cold information. C fibers are the smallest cells with unmyelinated axons called free nerve endings. They transmit pain, warm, and some cold information. Transduction is the process of converting a specific type of stimulus, such as tissue damage or inflammation, via specific receptors into action potentials or graded potentials. These receptors come in 2 main groups. The first is ligand-gated ion channels that open in response to specific ligands released with tissue damage that elicit fast responses. The second are metabotropic G-protein coupled receptors, GPCRs, which elicit slower responses through activation of G proteins. Here is an example of a GPCR that upon agonist binding allows for exchange of GTP for GDP and release of the G-alpha protein from G-beta gamma subunits. G-alpha subunits can bind to membrane-bound and cytoplasmic adenyl cyclase, kinases, and phosphatases, while G-beta gamma subunits are typically membrane-delimited and act on ion channels. In this way, signals can be amplified. Activation of ion channels, either directly through ligand-gated ion channels or indirectly through GPCRs, elicit small depolarizations called generator potentials. If enough of these channels are activated along the peripheral axon, the generator potentials summate and produce a large enough depolarization to activate voltage-gated sodium channels to produce an action potential that will travel from the periphery to the central nervous system. Since action potentials are all or none, that is, action potentials are identical after threshold has been reached, intensity of stimuli is encoded by frequency of the action potentials. Up to this point, we have been discussing how painful stimuli are encoded by neurons in the periphery. Adaptations in this signaling are involved in the long-lasting pain that is felt near the site of injury, known as peripheral sensitization. These types of pain are thought to be adaptive, allowing the individual to rest and favor the injured site. Allodynia refers to pain that is initiated by normally innocuous stimuli, such as touch or brush along the skin. Hyperalgesia refers to an enhanced response to noxious stimuli. These processes are mediated by substances released by tissue damage, such as histamine, serotonin, nerve growth factor, etc. These are collectively known as inflammatory soup. Peripheral endings contribute to the sustained sensitization via releasing substances that increase extravasation of blood vessels and release of inflammatory mediators from immune cells. In terms of mechanisms of sensitization, these inflammatory mediators act to increase and or prolong activation of ligand-gated ion channels through molecular modifications such as phosphorylation. Examples include GPCRs that phosphorylate ion channels, as shown here after stimulation of adenyl cyclase activity. Increases in intracellular calcium from flux through ligand-gated ion channels can stimulate calcium-dependent kinases such as protein kinase C that phosphorylate GPCRs or other ion channels. These molecular mechanisms allow for amplification and long-term sensitization of pain signals in peripheral tissues. Central mechanisms are also important contributors to prolonged chronic pain states. In the central synapse, primary afferents release glutamate and neuropeptides such as substance P. Presynaptic mechanisms can lead to increased neurotransmitter release. On the postsynaptic side in the spinal cord, or trigeminal nucleus in terms of trigeminal afferents, NMDA receptors play a key role in sensitization. Their activation leads to increased intracellular calcium levels that activate several kinase signaling pathways and transcription factors, ultimately resulting in regulation of gene transcription. Additional adaptations lead to changes in chloride regulation that switch inhibitory actions of GABA and glycine receptors to excitatory actions, termed disinhibition. Finally, there is more recent evidence that microglial activation contributes to chronic pain states. They release factors that contribute to both immune responses and adaptations in the postsynaptic cell that result in disinhibition. Together, these complex adaptations make chronic pain a challenging problem to solve. In summary, this lecture has highlighted the fact that pain is an individual experience that is influenced to varying degrees by biological, psychological, and social factors through processing by the brain. Specialized proteins transduce nociceptive stimuli. Sensitization is initiated by tissue damage through the release of myriad substances collectively known as inflammatory soup. And finally, multiple redundant processes in the periphery and in the central nervous system contribute to pain sensitization, increasing the complexity for development of novel pain therapies. For further details on the molecular basis of nociception, please refer to the text of the chapter.